A very warm good morning to one and all present here. On behalf of the entire Indian Botanical Society, I, Shweta Sharma, welcome you all to the IBS Foundation Day lecture organized jointly by Indian Botanical Society and CSIR National Botanical Research Institute. We are incredibly fortunate to have such a distinguished guest with us today. Please join me in welcoming our chief guest, Professor Akhilesh Kumar Tyagi, Senior Professor Retired, and MN Sahar Distinguished Professor Nasi to the stage. Along with him, I invite Dr. Ajit Kumar Shashne, Director CSIR-NBRI, and Dr. UC Lavanya, President IBS, to grace us with their presence on the dais. Sir, please. Please join me in welcoming our chief guest on the stage. Now I would request our director, sir, to welcome our chief guest with a bouquet of flowers. Now I would request our distinguished dignitaries to please come forward to light the lamp. I also request Poshali to please recite Saraswati Vandana.
या कुन्देन्दु तुषारहार धवला या शुभ्रवस्त्राभृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवैः सदा वन्दिता क्षमां पातु सरस्वती भगवती निःशेषजडापहा.
Thank you, Poshali. Now I would like to invite our director, sir, to please give us the welcome address.
A very good morning to everyone. Most respected Professor Akhilesh Tyagi, Dr. Lavanya, Dr. Sane, Dr. Tyagi, Madam Sesu Lavanya, all other dignitaries from maybe different part of Lucknow, my colleagues, all other people from other institutes, students, and all the staff of NBRI. It's a wonderful morning today that we have along with us Professor Tyagi. And who doesn't know Professor Tyagi in India? As sir told, he started coming here from the time of Dr. Sane, and we are really grateful to you, sir, that you are here again among us and inspiring us today. And you took out your time, came here, and we will be grateful to you, sir. And all other dignitaries, Dr. Sane, Dr. Tyagi, Dr. Lal, Dr. Lavanya, Dr. Sesu Lavanya, and all other people here. I welcome you all. Sir, this is a historical place. I always say that. I cannot stay without mentioning this thing again. This NBRI is a place of, you have seen the statue in the Udadevi Jora. So she is a lady, she fought with the Britishers and killed 32 soldiers, and that particular banyan tree is there inside the garden. And yesterday in NBRI, we have made a garden, that is Indian Heritage Garden in Banthra. So that garden contains different trees, the original trees, saplings of original tree or cuttings of original tree, painstakingly collected by the scientists of NBRI, rooted them, and these are the original replica of those trees which are being grown, planted yesterday, day before yesterday in Banthra with QR code, so that you can listen. If you scan the QR code, you can listen the history of that plant. So we have made for the North India. Now we will be making a replica, same replica for the South Indian trucks. Now we have filled up with First War of Independence, Champaran Revolution, and Dandi March. Gradually we will be filling up the track with different historical plants. So you are welcome to this institute. Again, I welcome you, and we are grateful to you that you have agreed to come here, and we are all inspired, and you will be inspiring all the students during the course of time of your lecture. Thank you very much, sir. And as always, Dr. Sane, he is my senior guide of this institute. So I welcome you, sir, here for your presence, gracious presence. Again, Dr. Tyagi, Dr. Lal, and all other dignitaries here, I welcome you all in this particular function. My staff, my colleagues, you are also welcome. Students, these are the pillars of NBRI. Thank you very much. Jai Hind, Jai Bharat.
Thank you, sir. Next, I invite Dr. UC Lavanya, President IBS, to brief us about the IBS Foundation Day lecture and formally introduce to us our chief guest of the day.
I forgot to welcome our director Seema. Sir, just a minute. Because I thought you are from NBRI. Just after coming here, I am saying, I am just remembering. Sorry for that. So you are of your institute, so you are.
Professor Tyagi, Dr. Shasni, senior student audience, Professor Sane sir, my senior Dr. Tyagi, Director CMAP, Professor Trivedi, my colleague Dr. Lal, and my old friend Professor Bansal, who has been associated with Professor Tyagi at Meerut, colleagues and friends and well-wishers. First of all, allow me to express my deep sense of gratitude to the Director CMAP, Director NBRI, Dr. Shasni, for allowing us to hold this function. This institute has always been helpful to IBS and has organized two conferences, one conference, one hosted and another co-hosted. Also, this institute allowed so many functions to be done on the platform from IBS. Like we arranged a function for felicitating Professor S.K. Jain for a Lifetime Achievement Award, and one lecture in memory of Professor S.N. Chatterjee was organized here by delivered with Professor S.P. Singh of Garhwal University. And likewise, there have been many occasions. Also, the NBRI has been helping us in many ways. Now we have NBRI as a nodal library of IBS, which was earlier held at BHU. So I am indeed very grateful to you, sir, for giving an opportunity to the IBS in many ways. And then let me express my gratitude to Professor Tyagi, sir, Professor Akhilesh Tyagi, who has kindly agreed to accept our request to deliver this foundation lecture. Now, a few lines about foundation lecture. The society IBS was founded in 1920, and on the completion of 100 years, it was decided that we should annually hold a IBS Foundation Day lecture. This is the fourth lecture in the series, and this lecture is to be delivered by the live member of the society and a distinguished botanist. Earlier 3 lectures were held at Calicut, CFTRI, and CSIR University Meerut. This is the 4th in the series for which Professor Tyagi has come here and accepted our request. Now, this is about foundation lecture. Now, a brief about Professor Tyagi. I have personally known Professor Tyagi since 1978. When I visited the department at the University of Delhi, when I was also doing my PhD, it was Puja vacations of 1978, and perhaps he was new to the department. And this was a very unique arrangement that time, that the moment I went there and I was told that I was working with Professor A.K. Sharma, Professor Tyagi and his colleagues immediately brought me to Professor S.C. Maheshwari. And what he did was, I will not forget this, is a culture we should also try to emulate, that he gave me a chalk and duster. Go to the board, what are you doing? This was the way of training the students. So friends, this is an occasion where we should remember our mentors. Professor Tyagi comes from the legacy of legendary plant molecular biology to the country, rather I will say he was the founder, Professor S.C. Maheshwari, who was known for experimental embryologist, then known for anthroculture, and establishing the area of plant molecular biology in the country. He comes from that legacy, and Professor Maheshwari found such a strong foundation that not one, he produced series of leading plant botanists from his department, Professor Tyagi, Professor Puri, Professor Khurana, and Mrs. Khurana, like many others. This is one department, I think, one professor in his lifetime has produced so many stalwarts. I don't think many of the scientists in the country can boast of that. They may have produced 1 or 2 students who have really made a mark in the international arena. This is one group Professor Maheshwari is remembered. And Professor Maheshwari was also very closely associated with society. He had delivered lectures on several occasions, and just before his demise, he delivered the lifetime attachment lecture at Patiala. So this is an occasion where I remember my seniors and elders. So now a few things about Professor Tyagi. The Secretary of the Society will read the citation about his achievements, but I give a brief introduction about other things. Although all of you know very well about Professor Tyagi, he is a fellow of all the major science academies in the country, as well as the Third World Academy of Sciences. He has a brilliant academic record that made him an entry in the Department of Botany, University of Delhi, in the laboratory of Professor S.C. Maheshwari, who was so much in demand those days. He obtained his PhD in botany from University of Delhi for his studies on haploid cell cultures and genetics, and then worked in Germany on photosynthesis related to chloroplastic and nuclear genes. He joined the Department of Plant Molecular Biology, University of Delhi South Campus, New Delhi in 1988, and became professor in 1994. He has served as head of Department of Plant Molecular Biology 1988, 92, 95, 98 to 2001, Chairman Board of Research Studies, Interdisciplinary and Applied Sciences 95, 2003, Director Interdisciplinary Center of Plant Genomics 2005 and 9, and Director of NIPGR from 2009 to 2016, and then now a senior professor, and currently Professor Meghna Saha, Distinguished Professor of Nasi at University of Delhi. He is among the few Indian plant scientists who have ventured to bring together knowledge of molecular biology, molecular physiology, genomics, transgenics to study, analyze and improve growth and yield. Major such interest of his group relate to photosynthesis, phototransduction, stress, reproductive biology, genome analysis, and genetic enhancement. He has been coordinator, team leader of major such efforts in India, has taken a plant science, which includes Center of Plant Biology at South Campus, Indian Institute for Rice Genome Sequencing, Indian Institute on Tomato Genome Sequencing, Network Project of Rice Functional Genomics, Center of Excellence Biotechnology, and Next Generation Challenge Sequencing in program in chickpea and genomics. He has published over 300 publications in journals, books of high repute, and has supervised of several postgraduate and PhD students. Dr. Tyagi has delivered over 250 invited lectures and chaired over 30 sessions in national and international meetings. For his contributions, the country has recognized him with different positions and asked him to help the growth of science in the country. He has been General Secretary as well as the President of NASI and also the Vice President of INSA. these are the very coveted positions in the country, and recognition to any scientist of this country. Vice President, Society of Plant Biochemistry, Plant Biotechnology, as well as Chairman of UGDBT Task Force on Human Resource Development, and has served as Chairman of PSc in Plant Sciences of DST. He has also served as organizing committee in several national interest symposia. In addition, he is serving on editorial board of journals like Transgenic Research, Morphogenetics, Genomics, RICE, International Journal of Plant Genomics, PEED Journal, and Journal of Plant Biochemistry Biotechnology. So these are the few things which I just mentioned brief. You can go on speaking days and days, but these are the brief things I just mentioned about him, the way he has been recognized for his contributions. So with this brief introduction, I will call it a brief because so many things could have been said. So this is all I wanted to say about you. So you can ask the question.
Thank you, sir. I'm sure after hearing such an introduction, we are all very much waiting for sir's lecture. So without further ado, I now invite Professor Tyagi to please come on the dais and deliver his talk.
इसका वो है हाँ मुझे Honorable Dr. Sane, Dr. Lavanya, Dr. Sasne, Dr. Trivedi, Dr. Sheshu Lavanya, Dr. Tyagi, my dear colleagues and friends. This function which is being held today is special in the sense that we are representing here today, all of us, one of the oldest society in the area of plant biology. As it was told to you in 1920, this was started after a meeting of stalwarts of the time at Indian Science Congress. And then you take any big name in the plant sciences, they have been associated. I just mentioned a few, and they are Dr. Sahni from Lucknow, Dr. P. Maheshwari, Dr. B.P. Paul, and obviously there are several large number of persons, and now of course Dr. Lavanya is the president. Coming to the institute, my association of this is now, as I said, started in early 1990s when Dr. Sane was director of the institute. And you see, sometimes little words of the senior people become very important. And I remember, maybe you will not remember it. One day I was standing after my lecture in JNU. Dr. Sane walked nicely to me and said, okay, it was nice. For how much time you were in Germany? I said, 2 years. So he said, I thought you were for 5-6 years after reading your papers. So in a way, indirectly, he said that he likes the work and he has been reading my paper, and these things become very important in the life of persons like us who are following the path of academics, where these words are the rewards in themselves. So that is the kind of association I had, and many of young people sitting here, Dr. Sawant, Anirudh, and all that, they were doing PhD at that time. So that is the time when I started to come to the institute, Prabodh was also there. I will now take Prabodh rather than telling Dr. Trivedi. So that has been my association with this institute. And then at the Department of Plant Molecular Biology, we moved on in the area of photosynthesis. That is where was the overlap with Dr. Sane's interest. But after some time, that came a little bit down, although when I work on stress, I still go to photosynthesis to find out that plants are doing well or not, but the basic work is, started to go down and new branches started to come up. And one of the branches that we have been following in our laboratory is the rice genome and gene function, and about that I will give you a little bit of the glimpse of one of the area that we have been following. Now, in this particular line, I would like to begin for the sake of students that genomics is not sequencing. Sequencing is the first step in genomics. The purpose is to establish the location of all genes and regulatory elements, and therefore, you keep on improving the annotation of the sequence which you have developed. Then you have to move to function of all the genes and regulatory sequences, and even then you don't know which is the better allele out of the two alleles present in the plant. So, you have to know that which of the alleles in a plant or large number of alleles in a population, which of them is important. And then you use this knowledge to the benefit of agriculture, health and the ecosystem. So, this all comes as a part of the word which we call as genomics. That is how is my understanding. Now, in the very beginning, when India was looking to sequence the genome, we had the opportunity to participate in an international program on rice genome sequencing, and that followed our participation in tomato and also in the wheat genome sequencing. So, we had some of the experience by way of participation in international programs, and then very soon Indian labs started to sequence the whole genome by themselves. We ourselves at NIPGR participated in sequencing of the desi kabli and the wild type of the chickpea, and other laboratories including your own institute became a leader in this particular area of genome sequencing and so on and so forth. Now, one of the first thing that we learned after sequencing rice genome is that rice genome is duplicated, and this is true for most of the genomes. And as you see here from the yellow strips, there are about 60% of the genome like chromosome 2 and chromosome 6 have got a overlap, and this is based on the concept of synteny and the presence of the genes in these duplicated regions. We also were able to predict that there are about 30,000 genes in rice, slightly more than that of Arabidopsis, and we could find that these genes could be classified into few major categories. For example, there are genes which are non-plant type, in the sense that they are in our body also, and this gives the idea that either we are vegetables or the rice is non-vegetarian food. But then there are others which are plant specific, flowering plant specific, monocot specific, and those which are specific to the genus Oryza or to Arabidopsis. So, in addition to that, at that time itself we reported about 18,000 SSR markers class 1 and 80,000 SNPs and different kind of polymorphism between Indica and Japonica. So, that was the start point of the genome. But what has happened over the years, that sequencing has improved, technology has improved, and it is possible to sequence large number of the accessions of any plant. So, Chinese sequenced 33,000 rice genomes, and a paper came in 2005. Now, when people started to analyze that, the concept of pangenome emerged, which tells that there are some genes which are common to all the varieties, and that is the reference genome. In addition to that, there are certain genes which are variety specific or accession specific. Therefore, whole genome of any plant is much more than the reference genome itself, and it may have several genes of utility. So, what we did not sequence several thousand, we sequenced only about 400 of the genomes of rice in India, but we used the data from about 5,000 accessions together, and considered out of them about 50 million SNPs, and after filtering all those SNPs through a particular pipe, we were able to get 80,000 SNPs, which are represented about 25% in the extra genome, that is the plant genome, and about 75% from the reference genome. And these 80,000 were used to develop a chip. I know that this department, this institute has also developed a chip of Gossypium. I keep on reading their papers, interesting work emanating from here, and these were tested for different purposes, like how much genome coverage is there, can we use it for GWAS, can we use it for mapping, can we use it for understanding variability. All these concepts were developed. We also used it as an example for GWAS, and in this case, as you see, we looked for the genes which are responsible for seed development, in which grain length, grain width, length to width ratio and 1000 grain weight were taken, and about 42 of the QTLs were identified. Now, out of these 42 QTLs, we found that there are about 36 in the reference genome, but there are another 6 which are not present in Nippon-Bare genome, which is the reference genome, but in the pan genome component of the rice genome. And in this, we were able to identify many potential genes by genetic mapping, which are controlling grain length and grain width. So, you can understand, just for your information, I would like to tell you that the gene for water submergence and also the gene for phosphate utilization is not present in the reference genome. It has been found to be present in specific varieties, and therefore, you can see the utility of pan genome in relation to that of the application of the information for public good. Now, next step that we took, we were experimental biologists, and therefore, we took the analysis of the transcriptome, and in this case, we were able to find the expression of about 20,000 genes, 28,000 genes expressing in different organs or in stress condition. And as you see in the green picture there, you can see the genes which are specifically expressing in seed, and therefore, it was possible to get information that which gene is belonging to which part of the plant or to which physiological state of the plant. Having done that, we over the time provided to the world 4 versions of annotation, and it is still going on. So, it is a continuous process, and we looked for phylogeny and expression pattern of several of the transcription factor families, epigenome regulation, regulatory network, and provided transcriptome of water deficit response, hormone response, and reproduction. One of the interesting thing which we found out of it is that there are in the cell many biological complexes which are multi-subunit, multiple proteins come together. Now, these proteins are changing over the time of evolution, but the changes are such that they compensate each other. If there is one modification in one polypeptide and it brings a structural change, there is a compensatory change in the other polypeptide, so that the complex remains intact and remains function, and that is the kind of changes we were able to observe by looking at the structure and the phylogeny of these particular genes. Now, once we get this differential expression of the genes, it was very easy to find out the promoters of different genes, and here in this picture you see the genes which are expressing only in anther and in pollen grains and not anywhere else, and neither in the seed nor in the stress stages or in the leaf and the root. And this provided us the regulatory sequences which we could evaluate to prove that function is really related to the promoter or there are some post-transcriptional components involved in this particular process. Now, this kind of knowledge has also been utilized for producing some of the components of utility. For example, in this case, one anther-specific promoter has been hooked to the gene for ornithinase, and ornithinase is able to convert the acetyl phosphothiazine into phosphothiazine, which is now a herbicide. So, what you do is that you put this gene under the control of a anther-specific promoter, so that nowhere in the plant there is deleterious effect. But in the anther, if you grow without acetyl phosphothiazine, then there is normal growth of female and male organs. But as you see in the lowermost blue color, if phosphinothiazine is produced, because the activity of the ornithinase under the control of a anther-specific promoter, then all the pollen die. And this kind of male sterile lines are also of utility for heterosis. And I again say, you see, we are probably growing in several things together, that again this institute has also been working on the male sterility. I think recently there is a paper in this particular area from this institute also in case of Gossypium. Now, we move further, and I will take you to some of the basic science, that in the regulation we get promoters, that is true, but promoters work because there are transacting factors. And most of the times we are told that it is the regulatory transcription factor which is responsible for particular expression, whereas there are other components like mediator and co-activators, and also there are general transcription factors, and we blindly take them that they might be doing the job in every organ, and therefore their phenotype would be responsible for changes in the overall plant rather than any specific organ or any specific condition. So, these three components ultimately tell the RNA polymerase that how this should be able to work. Now, I will take example of each of these three components one subunit, although there are multi subunit components, which tells you that no, it is true that they might be responsible for the whole plant, but their specific phenotypes can be generated by modifying even components of mediator or that of the general transcription factors. Now, here is one example where we are taking helix loop helix gene, coding gene, and this overexpression of this leads to male sterility. And male sterility first is reflected in the fact that anthers do not dice. So, indehiscence is the phenotype which is provided by overexpression, and we realized that this is because anthers are not dicing, because there is effect on the septum and stomium. The degeneration which is required for dehiscence is not there, and we also found that endothecium secondary thickening is affected, as you see in the blue colored slide, and this is reflecting the fluorescence of the lignin in this particular case. We also found in this case that pollen grains are sterile, as you see here, and this we confirmed in T0, T1 and T3 generation, that this sterility is quite significant, only 5 to 10% of the pollen were viable in this case. Now, why this is happening? To know that, we started to look at the sections of the anther from the transgenic lines and also from the wild type, and we realized that pollen are getting non-functional at the mature pollen, almost up to the vacuolated pollen stage. Both the lines are developing pollen grains, but at the mature stage suddenly the pollen grains are collapsing, and that is resulting in the pollen male sterility in this particular case. To know this, what is the region? We did a tunnel assay, and in this you see here in the top panel, you see that there is a reddish color and there are yellow dots. Now, this shows the programmed cell death that is happening in the tapetal cell at the time of development of the pollen grain. But in the third lane you see that number of the yellow dots has increased in the transgenic lines, and this shows that degeneration of the tapetum was advanced in this case of overexpression, and because it was not taking place at the proper time, therefore pollen development was not normal in this particular case. Now, interestingly, we found to our surprise, and this is for the students where sometimes you face the problems which look unsolvable, but there are answers for that. So, by over expression we found male sterility, and by down regulation also we found male sterility, and we were perplexed whether anybody will accept this work, and so we have to find out the reasons also for this. Fortunately, there is also other group working, and there is a mutant available, in which case for this BHLH gene, when the tunnel assay was done, it was found that programmed cell death has been delayed. So, in overexpression it has been advanced and not happening at a proper time. So, anther sterility is there, but at the later stage in the down regulation it has been delayed, and again it is not at the normal point, and therefore, in both stages you are able to get the same phenotype in this particular case. Now, to know further which genes are involved, we looked at the tapetum degeneration and anther indicence by doing microarray analysis or transcriptome analysis, and we found that the genes which are changing in these two stages are largely different. There is very little overlap, and this tells us that this could be representing two different processes altogether, that we are able to see the indicence and we are also able to see the male sterility. And this was reflected further when we looked at genes specifically related with cell wall modification, carbohydrate metabolism, lipid metabolism, ROS and cell death, and there was differential expression at these two stages in which we have analyzed the transcriptome. Thus, in summary, we were able to say that pollen maturation is affected because of lipid carbohydrate metabolism and transport. Dehiscence is affected because of lignin metabolism and cell wall modification, and we also have knowledge that this helix loop helix is able to make dimers and works with other similar proteins like EAT1 and TDR. So, this is one example I have told you, which is typical, that transcription factors are regulating the phenotype of the plant. Now, we will take another example, and this comes from mediator. The mediator communicates between the transcription factor and RNA polymerase and is made up of about 30 polypeptides. In animals, it has been found to interact with several interesting regulators like glucocorticoid receptor, estrogen receptor, vitamin D3 receptor, heat shock factor, and so on. In plants, there is little work in Arabidopsis, and it has been found that some of the subunits do affect the specific phenotype, like some MED13 regulates embryo development. Now, you may wonder, this mediator would be required in all the cases, but it is possible that at different time it interacts with different transcription factors, and therefore, it is able to cause the differential phenotype because of the changes in the expression of the gene. Now, we did a JAWAS for the 50 genes for mediator which are available in rice, and we found that some of the genes are associating very well with the grain size trait, and one of them is mediator 14-1. Mediator 14-1, when it was compromised in expression, that is RNAi, we found that the leaves are small, as you see there. Here the large veins and the small veins are small. So, ultimately affecting the narrowing of the leaf. Also, the plant was small. We also looked at the reproductive shoot apical meristem, and we found that the reproductive shoot apical meristem as well as vegetative shoot apical meristem in case of the mediator knockdowns was smaller, and this was largely because of cell size and cell layers which are present in this particular part of the plant. This in turn resulted in the sterility and also caused a small panicle development, as you see there. So, that was the effect also in the panicle. We also found its effect in the seed, and seeds were smaller, and there was a specific phenotype that hull was open in this case along with the small size of the seed that we were able to see. This shows that this gene works as a positive regulator for these particular phenotypes, because its knocking down is creating the phenotype that we are observing. So, we also found similar kind of effect in case of the root, where the branching of the lateral roots was affected. Interestingly, when these plants were supplied with auxin IAA, the lateral branching was restored. So, there was involvement of auxin in this, and therefore, this auxin levels were also expected to be changed. So, to confirm that this is happening because of the change in the cell division, we looked at the cells of different stages, and we found that there is different in the cell numbers at the S stage and G2 stage, whereas in G1 stage the cell number is almost same both in the transgenic as well as in the wild type. Also, several cell division related genes were found to be differentially expressed like cyclin genes in this case, or kinase genes related with cell division. We also looked at the auxin effect, as we were seeing in the case of the root, and by localizing the auxin in the roots, as you see here, we found more fluorescence in case of case of wild type in comparison to that of the transgenic ones. So, auxin levels were affected. Not only that, the genes related with the biosynthesis, transport, and also regulation of the auxin related genes were found to be affected, as you see in these quantitative PCR analysis. Then we looked at the reasons why this single gene is providing so many type of the phenotype, and for that we interacted this particular protein with several of the transcription factors, because we expected that maybe in different places it is interacting with different transcription factors, thereby giving different phenotype for different organs that we are observing. To our very pleasant surprise, we found that this interacts with YABI, which is known to control the organ size. It interacts with TDR, which is known to control the panicle and pollen development, and it also interacts with MEDS29, which is known to control the seed development. This data from yeast to hybrid was confirmed by immunoprecipitation, and in all cases we could find that this protein is interacting with such kind of thing, thereby developing a model that at different stages of plant development the MED14.1 is interacting with YABI, MEDS and TDR, and ultimately communicating their information to that of the RNA polymerase, and therefore, despite being only a communicator, it becomes an important component. And if there are natural variations in this in the expression of this gene, naturally you can consider that the rice plants would have variability in their phenotype. Now, interestingly, this particular gene has got a brother in the rice genome, which we call as MED14.2, and if you see the phenotype, on one end plants are small, but in case of MED14.2 the plants were not showing that drastic change, they were almost same, and this gene is the major expressing gene as a matter of fact. So, we also raised the knockouts by CRISPR-Cas and also overexpression plants, and we found that while MEDS14.1 works as positive regulator for seed development, that means seed size goes down as we are doing the knockout. In case of MED14.2 we found opposite, in the sense that when we did knockout then the size increased, and when we saw the overexpression then the size decreased. So, you can see that how these two paralogs having some similar functions might be affecting the phenotype in a manner that we get the final output from this. Now, of course, we wanted to see that since there is little effect on the vegetative growth, what could be the reason for that, and we found when we did yeast to hybrid with MED14.2, we found that it does not interact with YABI, but it interacts with TDR and MEDS29, and that is why it is having major effect on those two stages and little effect in case of the vegetative growth that we were able to see in this particular case. So we of course have data that how does it interact with the plant hormones and so on, but I will change the gear and take you to the last example, and that is of a general transcription factor, that they are also equally important as far as characters of the plants are concerned. And this is one of the transcription factor which allows the RNA polymerase to come to the promoter along with that of the TF2D, which has got the Tata binding protein TBP. So this TF2B has got 5 genes. As a matter of fact, normally in our body we find only one gene for them. So if it goes naturally, it will become embryo lethal, but plants have somehow increased in the number of the genes in each gene family. That is what has been observed. So there are 5, but one of them which I am showing here is the one which is showing increased expression in case of later stages of seed development. So we first took this, and of course we looked at its promoter, and we found that it is active everywhere except in the carpel, where we could not see any staining of Gus, but otherwise it is expressing the gene in these organs, which is similar to that of the expression we are observing by doing the qPCR. Now interestingly, when we try to raise transgenic, this is another problem to students, but don't give this example to your teacher, okay. The example is that we try to raise knockouts of this and also knockdowns, and in that case there was no plant regenerated, whereas in the control number of the plants getting regenerated was normal. So our interpretation is that its knockdown or knockout is lethal, and therefore we have considered overexpression or induced expression of this particular gene in order to find out its function. And this is the example given here, that when we overexpress in the seedling growth, as you see here, it limits the seedling growth and also the seed germination in case of the plant rice. It also affects the height of the plant by reducing the number of the internodes, size of the size of the internodes, and therefore we thought that if size is small, maybe gibberellin is involved in this particular case. So we looked at the genes which are responsible for biosynthesis of GA in case of rice, and we found that several of the genes are overexpressing, whereas there are others which are not expressing. And here we faced another problem, because if they are overexpressing, we expect that gibberellins will be higher, and higher gibberellins will naturally give a different phenotype than what we are observing, and therefore there should be some other region in this, and therefore we looked at the signaling genes. Interestingly, even signaling genes were showing higher expression, and therefore we have to go further and look for the genes which are involved in deactivation. To our help, it came out that there are several genes which are involved in deactivation of the GA or limiting GA biosynthesis, transcription factors. They are also showing overexpression. That means both systems are getting upregulated in this, and it is the homeostasis which ultimately is of course establishing this. The real test of it will be to check for the GA levels, which we are doing as of now, but that is how sometimes the problems need to be resolved by doing a little bit more work. But if you will go and tell your teacher, I am not getting regeneration, so naturally there is problem, it is lethal. So that will not solve it. You have to do experiment to say to your teacher that really it is lethal to the system. Anyway, we went further and we found that this gene is also affecting the heading time. Rather, it is reducing heading time, as you can see, by 14 to 24 days. And then we looked at the genes which are involved in flowering, and it was found that rice flowering locus T related genes like HD3, RFT and FLT are overexpressed. So that was in line of explanation which we are getting in case of the early heading in this case. We also looked at the downstream gene to these FT locus, and we found that they are also showing expression, including transcription factors like MEDS14 and MEDS18 in this particular case. Later on, we went on to analyze the phenotype of the seeds, and we found that seeds are of the same size, and therefore one conclusion could be that it does not affect the seed. But a student was more interested in going in deep, and so he analyzed the protein content of these seeds, and it turned out that there is about 10% increase in the protein content of these transgenic lines, and this is because of changes in all subcategories of the protein. It is known that when protein contents are increased, these are largely, I would not say always, but largely they are related with reduction on starch, and we found that yes, there was reduction in starch to our disappointment to some extent, and then shining. So, we found that amylose content was also decreased. Now, amylose content is related with the the glycemic index, and so I said it will not be of use, but then I was told by my student showing the literature that the change is not so significant that the glycemic index will change from one category to other category. So, you can feel happy with this result also. Anyway, so we found that this is the effect which we are able to get. So, friends, I have tried to show you some of the part of the work that our laboratory has been doing over the years. The simple message which is there is that it is not only the regulatory factor, but the so called mediators and also the general transcription factors are worthy of looking at for finding out the suitable phenotypes, which will ultimately be able to tell us the function of large number of genes. The work is tremendous. I understand that in case of rice, only function of about 6,000–7,000 genes is known. There are still 30,000 genes. People used to think in Arabidopsis we will find out function of all the genes by a certain time. It did not happen. So, there is a lot to be done, but also lot to be planned in a wise way that we are able to reach that target. One day we would like to have the profile of the expression of each of the cell. We would like to have the function of each of the gene, and more than we would like to know the alleles which we are able to ultimately put to use for the human welfare. We keep on doing basic research in the name of application, but sometimes the utility comes in. Interestingly, this is the story of rice, but in case of chickpea that I told you, which is another plant on which we worked at NIPGR, we find that it is possible to use this knowledge to produce better varieties, but that would be a talk at some other time. Thank you very much. So, questions? Chair should come up. Yeah. Let us have the question, then you can comment on both questions. And yeah. There are chemicals, I am not remembering the name, but people do spray and make the sterility set in. You will be alive today? Yeah.
Sir, there are chemicals tap it out. The nutrition conversion don't go here. So that is one people have been trying chemical induction.
Chemical induction. But in any way, sir, I presented to you a system in which you can spray phosphenothricin and get male sterility. That would also be essentially chemical induced, but it is engineered male sterility. That's the only point.
So I will give you a reference because one of my teachers was doing like this. I have a very general question. I think we are the best at this to understand many things. Have you ever visualized the plant type of rice which will give you the best harvest index? Practically as a being a unique tag. These are the components. I know that the number of genes and constellation. You can have changes by keeping the intact many things. Constellation can be intact. Do you have?
You see, Dr. Saab, normally I limit myself as far as work is concerned to what I can do. But I think, and for that matter, I would like to say to you that plant ideal phenotype has been conceptualized by scientists coming together from worldwide, and that phenotype in which there are less number of tillers and it is not more number of tillers, less number of tillers, and the leaf is erect. So these are some of the characters of that particular ideal phenotype, so that photosynthetic harvesting is concerned. So that is what is the requirement. Now of course scientists have also thought that can they convert rice from C3 to C4, and worldwide lot of work has gone on. We have not been successful even now, but there is interesting information coming out, and maybe one day that will be achieved. So these possibilities are there. What we are doing is to find out the function of these targets, so that we can understand that this is possible, and so therefore when you are doing application related experiments, keep these things in mind. But I am not far away from that. That's what I did not tell you. In chickpea, we have been able to get and release a variety which is high in protein content. There is also a phenotype which is erect, so that mechanical harvesting is possible. So these are some phenotypes in the other plants which have been thought of and have been achieved also. So that's the one. Yeah. Now you see, I don't know even what is a idiotype. I know plant type. Yeah. Right. So as I said, I don't make complicated things. Yeah. Right. Yes. No, term is there, I know. In the back. Yes, बतलाइए. Yeah. Still there is half grain field or no grain. That's right. Right. It is still existing, but you must be also aware of the fact that there are some QTLs which have been identified which give large number of grains, in which spike filling is better. But the problem is still there and it is not yet solved. That is true. धन्यवाद. चलिए मैं नीचे चला जाऊँ. ये कह रहे हैं यहीं पे बैठ.
On behalf of everyone present here, I would like to thank Sir for such an enlightening and enriching talk. Next up, I would like to invite Professor U.C. Lavanya Ma'am, Secretary IBS, to come on the dais and read the citation I would also request our director, sir, to please felicitate our chief guest with a token of our appreciation.
Thank you. Namaskar. I believe everyone has enjoyed this Foundation Day lecture delivered by Professor Tyagi ji. On behalf of IBS, it gives me immense pleasure to read the citation. Professor Akhilesh Kumar Tyagi, FNA, FASC, FNASC, FNAS, FTWAS, Meghnad Saha Distinguished Professor, Nasi at the University of Delhi, formerly Senior Professor, University of Delhi, Director, National Institute of Plant Genome Research, Executive Director, National Agri-Food Biotechnology Institute, President, National Academy of Sciences, India. During the felicitation function and the lecture delivered at the CSIR-NBRI Lucknow on 21st of January 2025. Dr. Akhilesh Kumar Tyagi, better known as an institution builder, is credited for bringing together knowledge of molecular physiology, genomics, and transgenics to study, analyze, and improve crop growth and yield. Working in the area of crop genomics and biotechnology, he led for successful Indian initiatives on genome-wide sequencing in rice, tomato, and desi chickpea. This has heralded an era of high throughput genomics in India. Under his leadership, pioneering contributions were made to the area of transcriptomics as well as neo and subfunctionalization of regulatory gene families in plants. These gene families include transcription factors, signal transduction components, protein processing components, RNA interference associated proteins. Further, functional genomics, genetic mapping are being used to understand molecular basis of yield as well as protection from yield loss under drought. This work is highly relevant to understand dynamic aspects of the function of biological systems and also to generate appropriate options for genetic enhancement of crop plants. Overall, more than 300 publications of international repute have been generated. He has been given J.C. Bose National Fellowship Award, National Bioscience Award, Nasi Reliance Industries Platinum Jubilee Award in Biological Sciences, GM Modi Award for Innovative Science and Technology, Bhasin Award for Science and Technology, Birbal Sahni Medal of Indian Botanical Society, and B.P. Pal Memorial Award of Indian ISCA, among others. He was also the President of Indian Society of Plant Physiologists and Vice President, Indian National Science Academy. Congratulations, sir. Yes, he has attended the very first.
That encouragement is very much necessary. I was a student and I was given opportunity to give a talk. So that helps.
Now I would request Director Sir to please felicitate. Now I request Professor U.C. Lavanya ma'am to please deliver the vote of thanks.
It's okay. I'm Seshu Lavania.
Oh, sorry. I'm
It's
so
okay.
sorry.
So, on behalf of the Indian Botanical Society and on my own, it is my proud privilege to thank the director of this institute, Dr. A.K. Shasni, for so kindly accepting our invitation to hold fourth Foundation Day lecture of the Indian Botanical Society. So you and your institute, NBRI, has always been very generous to support the cause of this society, having hosted and co-hosted the two of the All India Botanical Conferences, organizing award, memorial lectures, and the society in past, as well as serving the nodal library of the society's journal. Your commitment and support for the cause of science is indeed deserves wholehearted appreciation from the botanical fraternity. It is my proud privilege to thank my senior, Professor A.K. Tyagi ji, for so kindly accepting our request to deliver this Foundation Day lecture at NBRI, Lucknow. I express my deep sense of gratitude to Dr. Sane sir, former Director CSIR-NBRI, Dr. Trivedi ji, Director CMAP, Dr. Tyagi ji, former Chief Scientist from CMAP, Dr. R.K. Lal, he's former Chief Scientist, CMAP, President, Indian Botanical Society, all the other scientists of NBRI, CMAP, the research scholars, other invitees, for sparing time to hear this rather very informative lecture and to learn from the distinguished speaker. I would like to thank the media persons for much required coverage. I would also like to acknowledge the team of administrative, HRD, and IT units, stage management, and support staff of this institute for ensuring smooth execution of fourth Foundation Day lecture of the Indian Botanical Society at NBRI. Thank you all once again. Jai Hind.
I request you all to please stand up for the national anthem.